Hello and welcome. Let's think about our DNA for a second. Imagine it's the ultimate instruction book, a manual with 3 times billion letters, detailing how to build a human being from the ground up. It's mind-bogglingly precise, and most of the time, it works perfectly. But in this explainer, we're going to look at those rare but really critical times when it doesn't.
So, what happens when there's a typo in that book? Sometimes it's just a tiny little misspelling, but other times, an entire chapter gets duplicated or maybe deleted altogether. So, that's what we're going to dive into, these genetic typos. We'll start with the biggest chromosomal errors and zoom all the way down to the tiniest single gene defects, and we'll see why finding them is so incredibly important.
Okay, let's start with the big picture. If our DNA is the instruction book, then our chromosomes, well, they're the chapters. We've got 23 pairs of them. And sometimes, during cell division, mistakes happen. You can end up with an extra copy of a chapter, or maybe a piece of one goes missing. Now, one of the most common of these large-scale errors is something called a trisomy, and it's pretty much what it sounds like, tri, for 3. Instead of the usual 2 copies of a chromosome, you get 3. You can just imagine how having an entire chapter of the instruction manual copied and pasted in twice could cause some serious confusion for the body's construction crew. So, which of these duplicated chapter errors do we see most often? Well, this chart lays it out perfectly. You can see right away that Down syndrome, which is trisomy 21, is by far the most frequent. After that comes Edwards syndrome, or trisomy 18, and then Patau syndrome, trisomy 13, which is the rarest of these 3. And here we can see how a different duplicated chapter leads to a totally different set of instructions. So for Down syndrome, the extra chromosome is number 21. For Edwards, it's 18, and that's often linked with a very distinct sign, clenched fists with overlapping fingers. And for Patau syndrome, which is trisomy 13, features like a cleft palate or even extra fingers, that's a condition called polydactyly, are common. Let's zoom in on Down syndrome for a moment, since it is the most common. That extra copy of chromosome 21 results in a very recognizable set of features, like a flattened facial profile and a single straight crease across the palm of the hand. But the effects go way deeper. It's very often associated with heart defects, blockages in the intestine, and also a much, much higher risk of developing Alzheimer's disease later in life. Okay, so we've talked about extra chapters, but what if there's a missing paragraph? And what if the instruction book has this bizarre rule where, for certain sections, it only reads the copy you got from your mom, or only the one from your dad? Well, this is where things get really fascinating. It's a concept called genomic imprinting. This right here is one of the most incredible examples in all of genetics. Just look at this. It's the exact same deletion on the exact same chromosome, number 15, but the result is completely different depending on which parent it came from. If the deletion is on the chromosome you got from your father, it leads to Prader-Willi syndrome, which is characterized by this constant, insatiable hunger. But if that same deletion is on the chromosome from your mother, you get Angelman syndrome, a totally different profile. The mnemonic SAIL helps you remember it. Seizures, ataxia, which is a problem with balance, intellectual disability, and this characteristic inappropriate laughter. Same deletion, 2 completely different syndromes. It's amazing. Deletions can also produce these really specific and sometimes very unexpected combinations of traits, and Williams syndrome is the perfect example of this. A tiny missing piece of chromosome 7 creates this really distinct physical and social profile. So the deletion in Williams syndrome actually includes the gene for elastin. That's the protein that gives our tissues stretchiness, which helps explain the heart problems and some of the distinctive facial features often described as elfin. But here's the part that is just so fascinating, the behavioral profile. Despite often having intellectual challenges, people with Williams syndrome can have incredible verbal skills and this profound, sometimes overwhelming sense of friendliness and empathy, even toward complete strangers. All right, so we've been talking about these huge errors, entire chapters getting duplicated or deleted. Now, we're going to zoom way, way in, down to the level of individual words and even letters. We're shifting from big chromosomal errors to tiny single gene mutations. And right away, we have a really crucial distinction to make. Remember how we said Down syndrome is the most common genetic cause of intellectual disability? Well, Fragile X is the most common inherited cause. This is a typo that can be passed down through the generations. The typo in Fragile X is a very specific kind. A 3-letter genetic sequence, CGG, gets repeated over and over and over again. It's like a stutter in the genetic code. This repetition ends up silencing a really important gene on the X chromosome, and that leads to features like a long face and a large jaw. It's also very strongly associated with autism and, in some cases, unfortunately, self-injurious behavior. Now, other single-gene typos don't really change the body's structure, but instead they break its internal chemical factories. We call these metabolic disorders. Basically, the instruction for one single crucial worker, an enzyme, is messed up, and without that enzyme, a specific chemical reaction just can't happen. That can cause a toxic substance to build up or a critical one to be missing. Tay-Sachs disease is a tragic but very clear example of this. It's just a devastating domino effect. It all starts with one faulty gene, which means a key enzyme, hexosaminidase A, is missing. Because that enzyme isn't there, a fatty substance called GM2 ganglioside starts to build up inside the brain's nerve cells. That buildup is toxic, and it systematically destroys the neurons, leading to this relentless and severe neurodegeneration. You know, some of these single typos have consequences that just ripple out across the whole body, affecting multiple different organ systems. And cystic fibrosis is probably the most well-known example of a single faulty instruction causing such widespread problems. The typo here is in a gene called CFTR. Think of this gene as the instruction for a tiny little gate on the surface of our cells. This gate controls how salt and water move in and out. But when that gate is broken, the mucus in the lungs and the pancreas becomes incredibly thick and sticky. This leads to chronic, damaging lung infections, and it also stops the pancreas from releasing digestive enzymes, which causes serious problems with absorbing nutrients from food. And the key to diagnosis is actually a simple sweat test, which measures the amount of salt. So, we've looked at big errors and small typos. We've seen entire chapters and single letters go wrong. But why does sorting all of this out matter so much? Why is getting that specific diagnosis so absolutely critical? Because a diagnosis is so much more than just a label on a chart. For a family that's facing a world of uncertainty, getting a specific diagnosis provides answers. It explains why this is happening to their child. It unlocks the right pathways for therapy and management. It connects them with support communities of other families on the same journey, and it helps them anticipate what challenges might lie ahead. It turns the terrifying unknown into the known. And that's really the crucial takeaway here. Every single time we identify and understand one of these genetic typos, we learn more about how the instruction book is supposed to work in the first place. By studying the errors, we learn about the correct text. And that fundamental knowledge is what paves the way for every new treatment and every new intervention. And that leaves us with one final thought to chew on. We have gotten incredibly good at reading this book and finding the typos. The next great frontier is learning how to edit it. As our ability to understand and maybe even modify the genetic code itself grows, the question isn't just about reading the book anymore, but about what new chapters we might one day be able to write. Thanks for watching.